Thank you very much for the introduction, and especially thank you to Professor Takazawa for inviting me to address this symposium. I've already learned a great deal about these topics of ethnicity and race here in Japan and in Asia that I'll take back with me to the United States to inform my colleagues of what's going on here. You can see from my title that my interest is in how categories of race have changed over time, but also in how the persistence of those categories is very much still with us. So there's a kind of a duality, both of emphasis upon change, but at the same time a concern with how strong racial thinking persists. So what I want to do to start off with, to show you how the continuity is here, an article was published just about 3 weeks ago in a scientific journal that begins to make the point. Here's a publication, early November, in Nature, and notice the concern. For the first time, it says, we have the full genome sequence of an Asian individual. Now, why is this newsworthy? Why would we publish an article in which we say for the first time there's an Asian? Well, it's because the 2 previous sequences that have been published have been about, one is James Watson. You've heard about him earlier in the day and earlier in your careers if you're in genetics. He was the co-founder, co-discoverer of the DNA. Back in 1954, the publication put him on the big map. Now, his full genome has been sequenced and published. And the same is true for Craig Venter. So we have here 2 white men whose full sequence has been published. It's in the public arena. So 3 weeks ago, we have the first Asian, and the press briefing is what I would call to your attention. In particular, it says that the reason why we're doing this is because there's a reference genome. A reference genome are the 2 white males. And now the full reason for my putting this in front of you is the language here. The different ethnicity. Well, not really. Being Asian is not ethnicity. Neither is being white. So here's a confusion in the scientific literature. An ethnic group is often, in the world in which we make these distinctions, the one in which there's a clear difference with language and culture and sometimes geography and residence, but not race. Race are now these huge categories called Asian or white or sub-Saharan African is sometimes included, but ethnicity, that's a very different category. So right away we see a problem. We're talking about race, but we shift grounds to ethnicity. And how can I be so sure? Well, because in the very same publication, Nature, volume 56, there's a second article. It's the sequence of a Yoruban. So now we have the big 3, all 3 racial groups that we have since about 1820 regarded as basic. But think about this for a moment. Why would we believe that a single Asian, a single sub-Saharan African, and 2 white men would throw some kind of light upon ethnic, quote, racial differences? It's because we have some deeply embedded idea that there must be these huge differences. If I told you we had differences between a Christian, a Buddhist, and a Muslim, that we sequenced their genome, you'd find that a bit strange. You'd say, well, there are many Christians, there are many Muslims, every group. Well, surely with 1.4 billion Chinese, you add that to the Japanese, the Koreans, Vietnamese, Laotians, Cambodians, why would we think that mapping an Asian gave us any information about racial or ethnic differences, unless we have a deeply embedded idea that this is what's really important, being Asian and having your genome sequenced. All the more dramatic is the fact that, as you heard earlier in the day, earlier this morning, you heard a presentation that emphasized how in the early period of the Human Genome Project, we had the language of we're all the same. In the first 10 years of the project, here was the mantra we heard over and over again. It doesn't matter whose genome you sequence. It can be an Aboriginal, a Laplander, it could be a Swede, an African, a Japanese person. We're all so much alike. It doesn't matter whose genome we sequence. That was the first 10 years of the project. And indeed, notice this language from the UNESCO Statement on Race. It went so far to say that the statement makes clear that this concept of the biology of race has no legitimate place in biological science. That's as definitive as one can be. So what happened? Why in the last 10 to 15 years have we turned it around so that now we publish an article saying we have an Asian and 2 whites and a black? Well, again, early this morning, this is somewhat of an overlap. Something occurred in the year 2000, right at the same time that Craig Venter and the Human Genome Project's director, Francis Collins, got together at the White House and announced that they had sequenced the first draft of the map of humans. They concluded in this famous press conference, there's one thing we know. At the DNA level, there's only one race, the human race. That was in the year 2000. That same year, the US government passes an act. It's designed to address health disparities between the different ethnic and racial groups. And so you notice the language, health disparities, and in particular, it directed all funding agencies giving money to scientists. It directed them to ask these scientists, even to require these scientists, to publish their results showing differences between ethnic and racial groups. The goal was benign. It was in behalf of good things, namely, if we know more about how cancer rates differ between Asians and blacks and whites, we may be able to have some kind of programs which particularly focus on those issues. But that's not the story. The story is, once you tell people to report their data out based upon ethnic racial differences, and it's a requirement of the government, that's what they'll do. And indeed, in this 2005 Journal of Chest Physicians, notice this language, that black asthmatics displayed significantly different responsiveness than did white asthmatics. And in particular, the conclusion was that black asthmatics require greater concentrations of a particular corticoid in vitro to suppress activation. That blacks have, now notice here, blacks have a racial predisposition. Now so far, racial isn't genetic. But here comes the magic. Suddenly, this racial difference has been transposed into something called an inherent predisposition to overexpose for these pro-inflammatory cytokines compared to whites. So here we have it just a few years later from the Human Genome Project, the statement that blacks and whites are different in respect to their responsiveness. Now this letter was an approval from the government to the company called Merck for a new drug. And notice in this letter, just 3 years after the famous conference, notice that they said, this new drug reduces the risk of stroke, but it doesn't apply to black people. Extraordinary for a country which just 3 years earlier it said it's not about race. And now notice here, in this summary issue, this is the annual review of toxicology and pharmacology. An annual review is one in which you summarize the literature of a full year. So I'm not being specific in picking articles which make my point. This is a summary distillation of a full year of research. And look at what it tells you. We're going to get a better understanding of the molecular basis underlying ethnic differences in drug metabolism. And the purpose, of course, is ultimately to find differences upon individuals. But before we get there, we're going to talk about ethnicity. Again, white patients are not an ethnic group. That's a racial category. Whites require higher warfarin doses than Asians. And now Chinese patients require lower average maintenance of warfarin than whites. So now we're comparing a national group, Chinese, to a racial population, whites. The literature is confused on this topic. But what's clear is that people are starting to reinscribe, reinterpret, reinfuse the whole literature of science with the language of race. At a press conference just a few years after the Genome Project was announced in its first draft, this company's president called a press conference. And the reference group was, once again, Craig Venter and Celera Genomics. That was the reference group. And the claim was that Macrogen had successfully deciphered the genetic information of an ethnic Korean. Once again, notice the kind of mesmerizing concern, kind of really deeply embedded idea that somehow we could get to the ethnic and racial basis for distinguishing between Koreans and non-Koreans. In the interest of time, I'm going to skip through the Cartwright material just to tell you that in 1850, in order to justify slavery, medical doctors and medical journals began to publish articles suggesting that the reason why black people have a certain problem is that, for once, they consume less oxygen than white people, and that these differences are based on the structure of the body. Indeed, later on, there's going to be work on lung capacity in the United States and racial differences. In the interest of time, I simply wanted to show you this as one of the bases for late 19th century research on the topic of ethnic and racial differences. The cause that induces a Negro to run away is a disease of the mind. With proper medical advice, followed by those who practice this thing called enslaving people, that this habit of Negroes running away can be prevented. So now we've medicalized escape. We've turned people running away from slavery into a disease of the mind that only those who are medical professionals can come to terms with it and help prevent it. In the 1930s, here was another dramatic moment where science, medicine, biology come together with social issues. Now, here's a situation in which a very respected researcher deliberately infused patients in Puerto Rico with cancer, a deliberate infusion with cancer. Now, the person who did this had a certain attitude towards his subjects. In a letter, which will later on be the basis of a criminal investigation, Rhodes had this to say about his Puerto Rican subjects. What the island needs is not a public health work, but a tidal wave of something to totally exterminate the population. The chief medical researcher, funded by the Rockefeller Institute, infusing people with cancer in behalf of trying to understand, of course, how cancer works. But notice the mental set. Now, is this person an aberrant? Is this person a deviant? Well, take a look at what happens to him in his career. There was a criminal investigation, but it exonerated Rhodes. Many of his patients died, of course, because they had cancer. And the prosecutor wound up saying, well, he's a man of few scruples, but he doesn't deserve to go to prison. He was, after all, trying to learn about cancer in behalf of saving other people. And now I come to the social political framework of this development, namely that Dr. Rhodes went on to establish chemical warfare labs during World War II in Maryland, Utah, and the Panama Canal Zone. And really vital to my story about this man of few scruples was that he was given the Legion of Merit of the United States. This man who infused cancer into people got the Legion of Merit, and later on was appointed to the Atomic Energy Commission. More on the Puerto Rican issue with respect to ethnicity and science, the birth control pill. It turns out that in the early part of the 1950s, researchers from the United States went down to Puerto Rico, and they offered women this thing that they said seemed too good to be true, that they'd give you a tiny pill to keep you from becoming pregnant. And this woman, only 4 years ago, recalled what happened. She said, we all jumped with joy. We didn't even think about the implications. We were told this medicine was something that would keep us from having a child that we could not support. Of course we would be enthusiastic. And they were. Now we've begun to look back at this period, because people are going to talk about it, and they have. In the 9 years of this veiled research, what was happening was that unbeknownst to these women, they were the unwitting pioneers of the modern sexual revolution. They were the ones who were the experimental subjects of the world's first birth control pill, a tablet that turns out to be 3 times more powerful than today's pill. Now, we knew this very soon thereafter. That is, we knew in the early 60s that the pill was having a really dramatic impact upon these women in Puerto Rico. And why do we know it? Because comparison groups were in Boston. And in Boston, they stopped the experiments because of the side effects. And in other parts of the country, it stopped. But in Puerto Rico, it continued on for another several years until the pill was, quote, perfected. Perfected to be, therefore, legitimately used on white women. Herbert Spencer wrote a book back at the last part of the 19th century that I think is important for us to revisit. It was the most significant art book of its time. It sold more copies, 450,000 in hardback, than any book before or in the period of the next 15 years after. This was a book of great significance, and Spencer, following on Charles Darwin's notion of evolution, took the idea into the social cultural realm. And Spencer said that the fundamental basis of the continuum from savage to civilized was the development of the brain. And this he explained in terms of the way in which humans adapted to nature, in particular the seasons and the passage of time. He had an elaborated theory that primitive peoples had a sense of time relevant only to when birds migrate, or when fall or winter or spring begins. And thus, he had a notion that the more advanced and more civilized societies could encompass decades of thought, even centuries into their thinking, their planning, and their accumulation. As such, he believed that their brain capacity was vitally stimulated, and in these cultures, you could have a higher level of intellectual development. Spencer was such a powerful figure in the last part of the 19th, early 20th century that he was cited more than Marx. He was cited everywhere. The U.S. Supreme Court cited Spencer in several of its rulings, so much so that the Chief Justice once said to his colleagues, Herbert Spencer did not write the Constitution. So here is what happened with Spencer's work. And what I'm arguing here is that this hierarchy of cultures is still with us. It permits us to think about what happened with these women in Puerto Rico with the pill. It permits us to understand what happened with cancer insertion, the Tuskegee experiment in the United States on blacks with syphilis. All these things are understood in terms of the larger cultural framing of who is at the bottom, a primitive or a savage. Well, that was what Spencer said were the Aborigines. They were at the bottom of the hierarchy. The next came the Hottentots of Africa. They, he thought, were capable of annual thinking. They were still savages. Next came the nomadic peoples. Why? Because they could find ways of moving around the earth and understanding natural developments, but they could not accumulate. Nomads need to have very light travel, so they don't bring things with them. They can't accumulate. So he said the next level of civilization were people who were sedentary, people who could live on the land, at the water, and fish for a living, or hunt and gather and stay in one spot. They were sedentary. Next, however, comes the beginning of civilization. Next is the accumulation based upon surplus in food supply, surplus in agriculture, and that accumulation permits the possibility of people to develop cultures, city cultures, he argued. And at the very apex, there would be civilizations. At the very top, and I'm not exaggerating here, he put Oxford, Cambridge, Heidelberg, the Sorbonne. This is the apex of all culture and civilization. Now, this hierarchy of cultures, 19th century thought, is still with us. It explains much of what happened in the 20th and early 21st century. And now I want to take us back very, very briefly to what you already saw this morning with a different purpose. This is the famous double helix. I'm going to try to explain, in my view, how we got to where we are with the Human Genome Project, the HapMap, and a concern that turned from homogeneity, we're all alike, to a concern for difference, we're all different. This is the famous double helix, and you can see in the graphic, at the very base, it's split apart. Here's what they mean when they said, there's no such thing as race. In this room, any two people, chosen at random, but if I took your DNA, opened it up, split it, and put it on one of these Affymetrix chips, at every point for about 1,000 of these nucleotides, there'd be complete overlap. There'd be no difference. And then at about the 1,300th point, there'd be a single point of difference. It's called a single nucleotide polymorphism. You heard about it this morning. Then for the next 1,000 points, there'd be complete overlap. And then there'd be a single point of difference. In this room, any two people at random, or any two people in the world at random. Complete overlap in the DNA. Throughout the full 3 billion base pairs, there'd be complete overlap, except at these points of difference. So why map and sequence? Well, because this little difference might be in a region in which it was telling the protein to tell the cell to clot or unclot the blood when it reaches the open air. If it's, quote, a misspelling, that would mean that when the blood reaches the air, it doesn't clot, in which case the person has hemophilia. That one small difference could make a huge difference in your life, thus the notion that we need to map and sequence the whole genome. But notice, this is a very small part of the full picture. This is an actual genome slide, courtesy of Francis Collins' lab. And again, to repeat, any two people in this room, if I put your SNP pattern up here, would overlap except at these 3 points. Suddenly in the year 2000, things were going to change. Why? The very moment at which Craig Venter and Francis Collins and Bill Clinton and Blair come together to say it's the end of race, at that very moment, something's going to happen. There are 3 billion base pairs, but as you heard again this morning, there are between 3 and 10 million points of difference between any two people in this room or any two people in the world. Now the drama emerges, the plot thickens, because now, with new technology in 2000, IBM announces a new supercomputer to go into these chips, and what can it do? At 7.5 trillion per second calculations, analyze your DNA. Now everything's going to change. 99.9% alike, but that 0.1% difference is 3 million points of difference between any two people. And a computer, given the data of 10 people or 1,000 people or 7,000, and told to find differences, will find them. If I'm given a supercomputer program with a program called Structure, and I get 5,000 people from Chicago, 5 from Los Angeles, and 5 from Tokyo, I'll find extraordinary patterns of difference. Now the question is, what do I make of it? A new field will be born. It's called pharmacological genetics or pharmacogenomics. The new field, born at the very moment in which they're saying race is over, at that very moment, listen to this language. It says all the markers that we know today differ in frequency among ethnic and racial groups. So here's the contradiction. At the moment in which we say we're all the same, a new field emerges to say that no, we must look at these ethnic and racial differences, and it dictates that race be considered in studies aimed at discovering phenotypes, genotypes. Why? Because of disease risk, drug toxicity. Now, how would they know that in the year 2000? How would they know that these markers in the DNA differed by ethnicity and race? Where was the empirical basis of this assertion? The article doesn't tell you. Had there been work going on across the globe, across ethnic groups? It was all in the sense of, well, we'll get to that perhaps in the question and answer. Once again, this morning had a reference to this overlap. This is the famous drug that ultimately would be developed for African Americans, the first, what they call ethnic drug, actually the first racial drug. The original study, done in the early 1990s, was across all categories. Race was not the issue. And the purpose was to get this particular nitric oxide deficiency level up for those who had had heart failure. So that was the purpose. The trouble was, we already had drugs on the market, and so the FDA, Food and Drug Administration Advisory Panel, said no to Vital. They voted it down 9-3. But markets are important. And the company, one of the company's persons got together with others and said, let's go back and re-examine the data. And they re-examined it, and they found that blacks seemed to have a better response to this than whites did. Why? Well, about 5 million Americans suffer from heart failure. This proportion of them are black. Therefore, there's a higher ratio of blacks to whites who suffer heart disease. And you recall I mentioned racial disparity research, that the government had mandated that we keep records based upon racial disparities. Well, now it's going to come home to roost in the sense of an analysis of what's really going on between blacks and whites. This disparity can be attributed to many factors. Could be pathophysiology, primarily in black patients who have a nitric oxide deficiency. The company then releases these data, and it says that Bidil, this new drug, reduces mortality in 2/3 of African Americans, but it proved to be of no benefit to whites. And now the famous press release. African Americans are affected at a greater rate by heart failure than Caucasians. Indeed, African Americans between the ages of 45 and 64 are 2.5 times more likely to die from heart failure than Caucasians in the same age range. Fairly definitive, except for a little detail. Over 65, where almost all of the mortality occurs, 93.7%, that difference disappears. So what they did was to manage this particular statistical imagery here so that this age group, 45-64, it was true that 2.5 times more blacks than whites are dying or having heart failure problems, but over 93.7% of all the problems with mortality are in the age group in which the difference between races disappears. If there's one graphic which I'd like to emphasize and leave with you for this presentation, it's this one. This is the work of Richard Cooper and his colleagues. The same time that we're looking at Bidil, the same period this was being published, was a massive study of 8 nations looking at blacks and whites and Asians and looking at heart disease problems, hypertension prevalence rates by country. Now, if you stay inside the United States, you'll see that U.S. blacks and U.S. whites had very different rates of hypertension, that the black rate is higher than the white rate. But if you broaden your scope of your understanding across 8 nations, something rather dramatic and amazing happens. Namely, the country with the highest rate of hypertension is Germany, by a huge measure, and the lowest rate is Nigeria. Thus, if you stay inside your own country, the United States, you'll make an important and huge mistake in terms of the imputation of the causation of hypertension. You may think it's about race, namely maybe the biology of race or maybe the social forces of race, but you will not understand if you go outside your country that what's going on here can't be race at this level. It's got to be something else. It turns out that the largest ethnic group in the United States are Germans. So maybe when Germans move to the United States, their blood pressure goes down. Maybe when Nigerians or sub-Saharan Africans come to the United States, their blood pressure goes up. Maybe it's as simple as that. Where are we headed and why? Well, here's an article published, again, just 3 years ago. Pharmacogenetics and geographical ancestry, implications for drug development and global health. And here's what the article states. This is simply the reference for those of you who want to pursue this, and here's the language. There's a Pan-Asian SNP consortium. Scientists from China, India, Indonesia, Japan, Malaysia, the near Philippines, Singapore, Thailand, and Taiwan all get together. And in behalf of what? In behalf of the fact that other parts of the world, Iceland, Estonia, Sub-Saharan Africa, genomics, are talking about these patterns of diversity and drug applications. Maybe we need to have something for Asia. The Asian, you remember the first slide I showed you? We're going to now have an Asian issue to uncover the breadth of diversity and the extent of similarity with Asian populations. The purpose being to develop pharmaceuticals. Our increasing understanding of human genomic variation, specifically its application in pharmacogenetics, might shift our focus away from individual differences towards interpopulation differences. Now that's the opposite of the rhetoric of individual application of pharmacogenomics based upon your particularized genomic sequence. Let me conclude with some reflections on what I'll call the wrong questions in the debate about race. The current debate is very heated in the United States and in some parts of the scientific world. People say, yes, we should, or no, we shouldn't use race in scientific research, clinical medicine, and indeed in pharmacogenomics, pharmacotoxicology, and forensic science. That's part of the debate. We should or we shouldn't. That's not much of a debate. That's sort of like take your stand, take sides. Which side are you on? That debate is unproductive. It goes nowhere. I think that the right questions are to be reframed. The question should be, under what conditions do we use race in clinical medicine and scientific research? Under what conditions should we be wary and reluctant to deploy race in these arenas? That seems to me to be a question worthy of a debate, not the yes we should, no we shouldn't. One of the most famous studies in the world is this one. Highest rate of, where are we? The highest rate of hypertension recorded at this period was in North Korea and Finland. They reduced it by 73% in 2 decades. By what? You see. Nutrition. And here are some images with which I will leave you on right questions, wrong questions, when to use race in research. Let me, for the moment, I guess I'll take an extra 1 minute here before I get to this conclusion. If you find differences between whites and blacks with cancer, and we find them, prostate cancer in the United States, huge differences. Blacks have twice the rate of prostate cancer as do whites. And the question would be, what's a good amount of kind of research to pursue? Well, if you're interested in oncogenes, cancer genetics, you go right from the clinical population, black people, back to their genes, back to their genetic structure. And that, it seems to me, to be the wrong way. Why? Because of what I'm going to show you. The better way is talking about, well, maybe prostate cancer is a function of everything from cell development up through morphology, embryology, developmental biology, finally to the person. You finally have the notion of genotype to phenotype. That would be interesting research. So now we have twice the rate of colon cancer, but the average American gets 10 grams of fiber per day. Ugandans get 5 times the amount, and they have almost no colon cancer. Let's look at race, but look at it in the context of the way in which race shapes our social life, our culture, our existence. And here's another slide. In the United States, lots of research on diabetes and genetics. Go right from the diabetic back to the oncogene. Well, here's another way of looking at it. Turns out that in their traditional country, the Pima, who are kind of the poster children of high rates of diabetes, 23% prevalence rate of diabetes in cities. New Guinea, urban area, 37% diabetes. Aborigines, in their urban environments, high rates of diabetes. Throughout the whole world, it's true. But in their, quote, ordinary circumstances, almost no diabetes. Why would we think that genetics is the answer? So I end with my frame about right questions, wrong questions. It is possible to use race in research. It's indeed an important thing to do. But you do it in the course of a lifespan. How are people becoming more hypertense or more stressed or more likely to live around toxic waste dumps? Living around cell towers in which it might affect how your cognitive apparatus is developing? All these questions are important and useful, and the race of the people in those groups is systematic. It is in that context that I think we can reconsider using race in research. Thank you.